started. I'll start a new notebook.
Rissa, what was that website that you had up on Thursday last week that had the ketones and the aldehydes and the isomer pairs in PDE?
Yes. Sorry, I know what you were talking about. I'm trying to find the email because I thought I emailed myself the links. There we go. Yeah. Pulling it up. So you got it. Should I make an announcement with this or something? You're saying yes. Okay. Yeah, I like their nice pictures too. And this is another little cheat sheet. This one's a Chem Libre text under their alcohols and phenols chapter. But I like this because it kind of showed you, well, it put the reduce and oxidize terminology in a different way that we didn't really discuss in this class at the end. We had kind of discussed it in pieces with, like, you know, determining the oxidation state of, let's say, you know, carbon in this one where we looked at the sharing. And so in this molecule, right, carbon would have a -3 oxidation state from the method that we learned. So just assigning the electrons. But then we can see as it, you know, goes along and gets oxidized, so putting this in the context of the reactions we learned, that we can start with the primary alcohol and then oxidize it, so then it makes the aldehyde. And then, of course, if we had started with a secondary alcohol, we would make the ketone. So just comparing the oxidation states. And then, of course, most oxidize with our carboxylic acid. And then thinking back to our method, right, of oxidation state. So oxygen, of course, gets all the electrons in this case. And this one splits it, so that leaves carbon with a +3. If people follow, do we want to do the determining the oxidation states? Although it's not really going to be on your exam, but that was something that we did cover this quarter, the organic oxidation states. So how are we feeling for our exam? Yeah.
Are we going to have any of those, like the hydrolysis reactions and those kind of things on the exam?
Let's see. So looking at the exam, so I'll just kind of go through it initially. So it starts off with 5 multiple choice questions. And so there's some like writing nuclear equation questions, galvanic cell questions, coordination compound questions, drawing like the peptide chain given your amino acids and doing the linkage correctly. There's like naming the hybridization and an IUPAC naming, current, plating out, more coordination, naming, and ester naming. Ooh, this looks weird.
When you say naming the hybridizations, it would be like, so if you said name the hybridization of the propane and, or the carbon in the propane, like up there, is that what you mean?
Yeah. That's what I was trying to do. Yeah, so we could do like a hybridization question.
Can you read back that? So nuclear equations, galvanic cells, coordination, compounds, hybridization. I feel like I missed something.
Ester naming,
IUPAC, current plated.
and then there's also polymerization. Yeah, so I kind of laid off of the fats and the hydrolysis. So I don't know, were those the hydrolysis reactions you had questions over?
Yeah,
Or-
it was just on the lab that we did.
On which lab?
On the synthesis of aspirin.
Oh, so just how that comes together, or? So for the exam, what you should focus on, here, we'll come up with another hybridization in a second, but you want to focus on like how amino acids come together. So we can go through that again. So that would be one of our condensation reactions still. So making that amide bond. And so we could do a practice in a second. And then also like how polymers form. So again, looking at that connection, and you're just looking for that amide bond again, the nitrogen to that carbon of the carboxylic acid. And so you're always looking for that connecting point. Just looking through the exam. So yeah, so we said let's do some hybridization practice. And I'm going to have to move my drawing. And so for this one, I'll just highlight the carbons or nitrogens to do the hybridization on. So let's draw another carbon. All right, so identify the hybridization at each of those highlighted atoms. And so I'll give everyone a couple minutes to work on that, and I'll keep handing back papers. Anyone in the chat, by the way? Oh, I see you in the chat. So ask questions as well in the chat if you want me to go over something. Are we ready to start going over these? So what's the hybridization on this nitrogen? sp2. Everybody agree with that? Excellent. See some nods. What about this oxygen here? So don't forget, how many lone pairs does it have on it? Trick people. So we draw our lone pairs. So now what do we think it is? sp3. So don't forget, right, it still has that tetrahedral core to it, you know, accounting for those lone pairs. Let's see, let's look at, what about this carbon? sp2. And we could draw that hydrogen so we can see that a little better. What about this nitrogen? sp3, another tricky one, so drawing those lone pairs. Excellent. What about this carbon, our methyl carbon? What is it? sp3. Oops. Since it's attached, draw that better. So that's 3 hydrogens put into a carbon, so attached to 4 things. And then this last carbon here, what's its hybridization? sp2. It's a lot of sp2s on this one. All right, what are we feeling next? How do we feel about our galvanic cells? So let's do an example. Just make sure I don't pick the same metal. So sketch the galvanic cell. And then calculate the cell potential. And so let's try aluminum 3 plus, 1 molar solution, and copper 2 plus, 1 molar solution. Are we still working on this, or? Okay. I'm just gonna go and print the 20 amino acids for y'all so we can practice the amino acid drawing. Does anyone want to draw what they have for their galvanic cell? And you can either do it up front or if you want to do it on the far wall too. Nice wall, either one. It's a nice wall. Okay, I will copy. That looks good. good. 2 separate chambers. And I see the salt bridge, so that's essential.
That's it, right? Or you toss it right there.
I just asked for the picture. You want to go for the equation?
No problem.
Yeah, if someone else, so that looks good. Anybody have any criticisms of that? I'm not finding any myself. We have the flow of electrons, so I'm just copying it down now. So drawing in the voltmeter part, and so good, right, solid metals at each point. So aluminum, and then showing our ions in solution. That's excellent. Electron flow looks good. Copper. Perfect. And then, so does anyone want to write what the reduction potential is for us on the board? Writing out our half reactions. So copper. And so, yep, 1.66, 0.34. So do we all get 2 volts for that one? Excellent. And so just, we could calculate, right, more with this. So if we had asked then for delta G, we could have plugged it in using our cell potential. And then, right, note what we wrote on the board. So if we went further with this, right, our N, what would our N be for this case? So 6 electrons. So just note that had we gone further with that, our N would be 6 to have the same number of electrons backwards and forwards. Or, and then if we were calculating K, right, same thing, N would be 6 there. What is what? The F is a Faraday's constant. So it'd be this number for that equation. So let's do some more practice. So see if you can draw this amino acid chain of gly, met, take a few minutes to try to draw that, especially paying attention to the amide bond formation. And I will continue passing back papers. Where was it? Now, as you draw it out, does anyone want to volunteer to put it on the board? And tag team it too. go for it. You can even start off by just drawing the first two. If someone wants to do that.
Let's not let me write that yet, but you draw the last two.
Yeah.
I don't know if it's right.
Bueller.
I don't know if it's right.
So let's see. I haven't made my way. So the glycine looks good, right? So everybody drew that first part. Now we have our amide bond formation with methionine. So now I have to draw it and also look at yours. So 8 carbon there, so that looks good. And then you drew the whole sulfur chain. So it has 1, 2, and then a sulfur. So that looks exactly like what you drew on the board. Sulfur, CH3. And then just going back to this carbon. Then we have carboxylic acid carbon again. And we'll have new amide bond formation. So that brings us to asp.
Is there a hydrogen off of?
Oh,
Yeah, okay. So that was good.
yeah, so that was good. So who wants to try? So it'd be with aspartic acid. So you just have to draw. So it'd be going to that nitrogen.
So I don't love the book, but the book makes it a little bit easier to picture what the chains look like a little bit easier than notes, because those are really pretty pictures. But the chart in the chapter makes it easy to see how they all fit together.
Let me pull up that picture. Yahira, are you volunteering? No. You could totally go for it. I think Cassandra was volunteered. Voluntold. You should totally go for it.
I've already done a speech today. I've been impressed.
That means you've gotten it over with. The nerves, the
And
speed.
Marissa, if you scroll up just a little bit, like right there is where it has the amino acid base, and then like it changes for this where the R is, and that made it really easy for me to, yeah, right there, like made it easy for me to understand what was supposed to be going on with like multiple changes.
So you like this representation, or?
Well, the one on the left just shows you the base amino acid, and then all of the chains are down below it.
Oh, yeah, I see what you're saying and what makes sense to you. So maybe this will help people too. We look at it side by side. So if you look at this representation, all she's saying is the R group, oops, right? It doesn't matter what it is, whatever crazy thing is hanging off of it, like with methionine, right? So all that's just the R. So when you go to draw all these chains, if you kind of put it like that, that might be easier so you're not confused by the R group.
It was hard for me to figure out what direction they all needed to go in, and like how to draw them.
So that may be helpful. And then so that may help people see that you're just chaining, right, these nitrogens. So the first one we started with, right, glycine. So we just started that off with our amine there, and then took our carboxylic acid and got rid of the OH essentially, and then attached the next nitrogen to it. So I think that does kind of simplify it.
And I think there's actually something below the chart also that shows, yeah, right there, like kind of what it looks like once you chain them together.
I see what you mean with the R group on that orientation.
The pictures were really pretty, but they were hard for me to understand when I had to start adding in other stuff.
Yeah, I agree. More pictures, the better. So if we look at this as kind of our guide, so who wants to tack on ASP to it? Should I do the next one? Yeah. You might as well just go for it. I don't see anyone chomping at the bit. I would just watch. That's a minor criticism, but I think you attached histidine at the wrong. Yeah. So it kind of goes back to Nikki's hint, which now I'm realizing is kind of more helpful. Because you always have, right, your amide bond, so C double bond O nitrogen. And then, actually, if we go back. This one? Yeah, so you just have to drop a hydrogen there. Oh. Yeah, so that bond, right, that's okay. You just need one hydrogen because one went away. So that's good. And then there's always that carbon with the R group, like she was saying. And then another hydrogen, then your carboxylic acid. Or you can look at what I've drawn on the screen. I was trying to help. Yeah, I think you were there for the ASP one. My only complaint was that there was one hydrogen. show everything on screen. So then, right, if you're following in class too, so there's our aspartic acid, right? That's the nitrogen, but it's kind of easier with that textbook layout. So nitrogen, secondary carbon, carboxylic acid carbon. Then you put whatever group that was attached to it. So I just put an R for a placeholder, but now I can go back and rewrite it. Yes, that looks good. Then I think there's an extra carbon there, right? People following? I'm just gonna erase it.
I think that might be part of the chain, actually.
Did I get confused?
So the book says aspartic acid is CH2COOH.
Oh, I think it needs to be here. That's where the carbon needs to be. There we go. That fixes it. So that's what I have drawn there. So that's the aspartic. Now we make our last one, histidine. So C double bond O of aspartic makes our new amine connection. And so going back to the book representation, right, then you just continue along. So carboxylic acid, lose the OH, attach your next amino acid, right? And again, amino acids all have that basic form, so that's why you attach it to the nitrogen. So I'll pick a new color. So now nitrogen, they always have an H, then your carbon, then your carboxylic acid carbon, and so we can leave it as that form, and then your R group. So this has an H, this would be R, and then of course for histidine, that R should be just like Eve drew on there, if I could find it. There we go. So histidine with our 5-membered ring there.
Where is an appropriate place to show the waters that are formed?
This case isn't really asking for that, so that'd be more if you were doing like glycine plus met, and then you could like more explicitly draw the waters, but this one's just asking for like the chain. So it should look something like that. But now I like your general cheat sheet form, right? So N CHR. Then it would be the next one, NHCR2. And then you could go crazy forever.
The book doesn't do a lot of things right.
Yeah,
This was really.
that's good, yeah. I mean, that's why it's always good to look at different resources, because sometimes a book will have nice pictures, sometimes it gives you way too much math and makes you scared, but in this case, at least, it broke that down. And especially for OChem, I suggest that, look at different resources, different pictures, looking at things in different orientations might help. All right, let's go back. All right, what do we need to practice more? You want to do like high spin, low spin stuff? Do we need to review like the current plating problem? Like all those problems that involve like how much current is required to plate out, how many blahs? Okay, let's do one of those. So how much current is required to plate out 0.035 grams of, let's do aluminum from an aluminum 3+ solution in 10 minutes. How are we progressing? So where can we at least start with this? What do we know? How do we start this? Excellent. So let's start there so we can get moles right. They only gave us grams. And so let's just set up our dimensional analysis for now. And what's the molecular weight of aluminum? 26.92 grams. And so we could solve this and punch that in, that's fine. But then what do we want to go from there? So we have moles of aluminum. So once we get moles, excellent. So now let's do that. So 3 moles of electrons for every 1 mole of aluminum. So just setting up that dimensional analysis. Perfect. And so what happens once we have moles of electrons? What could we convert that to? Yep, and then how do we do that? All right, that looks excellent. So let's punch that in now. .982, okay. So 0.035 divided by our mass, and times it by 3. Again, since it's aluminum 3 plus going to aluminum 0. Plating out, so we need 3 electrons. And then our Coulomb conversion. So do we get like 375 coulombs roughly? Can round to 0.5. Double check my math, of course. And so now we just need to get current. So amps per coulomb. And we need to get our total number of seconds. So we can multiply 10 by 60 on bottom. I'm just writing out the conversion explicitly for everybody. And so what do we get for amps? So let's divide by our minutes, or seconds, excuse me. So I get like 0.63 amps roughly. So let's see some nines.
What's the last piece of information on mass?
Yeah, just because the amps is coulombs per second, so we had to convert minutes to seconds.
What was the answer?
And so we should have gotten 0.626 amps for our current. Let's see. Let's do one of these practices, oxidation number, coordination number, and name, or the following. We have an answer for this one. So oxidation number for that's not what I got, but I mean, I could make a mistake, but so just balancing the charges. So sodium, yeah, I like the answer of 2. And so let's finish running through that. So right, each sodium contributes 1 plus charge. And then bromines and chlorines are all -1. So we have +4, -6. So then that leaves us with a 2 plus missing on tungsten. And then what about the coordination number? So 6. And so that just goes back to how it's bracketed. So bracketed with our bromines and chlorines in the bracket. And so now how do we name it? Sodium tribromo, trichloro, and then what was the last part? Tungsten, oops. Tungsten, and then we just need to put the 2 at the end there so that we know how many sodiums that we needed to balance.
Why is it tungsten and not tungstonate?
Tungstonate would be fine.
Okay.
So kind of the key pieces with that is, right, we don't put tetrasodium, right, because we have the oxidation number of 2 for tungsten to tell us the sodiums. But then with the ligands attached, right, we did have to use the tri, otherwise we don't know. We don't know if it's 5 bromines and 1 chlorines, right, to give us -6, so we have to specify there for our ligands. Let's see. What other things do we need to look at?
Should we look at one of the nuclear equations? Is that like a half-life problem?
We have simple half-life problems of just like how much remains after so many half-lives. And then the other nuclear equation is just figuring out like what the products would be. So let me see if I can make one up. So let's say you have hafnium 192 and it decays to make 3 beta particles. And another daughter species. So identify the daughter and write out the full equation. So see if you can figure out who the unknown species is. I'm still working on it, need more time. So does anyone want to write it on the board for us? Yeah. Yeah, just go for it. We'll help you out. If you write it on the board, we'll all go through it together and help you though.
So hafnium 192 is atomic number 72. So I started with 92, 172,
So 192, 72, hafnium, perfect. And then, so the exam one might be a little trickier, but just read it. So in this case, right, this one's just decaying. So on your exam, there might be stuff adding to make something, just hint, hint. But and so what do we say? So it makes 3 beta particles. So what's the symbol for beta? So we had the little beta, but then what were the numbers? So 0, -1, and then we'll put a 3, so then know that you're multiplying, right? So 3 times -1 would be -3 on this side. So then what would we need? Let's just start with the bottom number. So what would we need on the bottom number to balance out that -3? So 75, and then, so now we have a 0 on top, so 3 times 0, still 0. So what's our top number? So still 192, except, so what's element 75 now? Yeah, rhenium. So Re. Excellent. All right, we're kind of running out of time, so I'll go through the exam just one more time of what the topics. So like I mentioned, so there's multiple choice and there's simple radioactive questions. So, you know, kind of doing it the simple way, like how many half-lives, what percentage remains type of question. So if you could set up the simple chart, you'll be in good shape. Oops. You know what I mean by that. So like 0 half-lives, you know, you have 100%, 1 half-life, 50%, you know, second half-life, 25, etc. So if you can kind of do that with percentages, fractions, you'll be okay. There's something about activity series, so giving you different metals, telling you how they react, can you order them? Talking about what the geometry has to be, if it has geometric and optical isomers, you may want to look at that. Let's see, multiple choice questions about DNA and some molecular formulas, so it's like simple alkane, alkene naming, so that sort of thing. Then, like I mentioned, the next section has 5 short answer questions. So one of those being the nuclear symbol ones, the galvanic cell question like we went over. Then there's the whole strong field, weak field type of thing with the splitting diagram. And of course, just figuring out electron configuration, all that. Diamagnetic, paramagnetic. So I think you'll be okay with that. You know, just show your work And it should be, it's an octahedral complex, so don't forget, right, what the splitting diagram looks like. So 2 on top, 3 on the bottom. And then just show your work as always. Then there's the amino acid naming one like we practiced. So being able to write that. Hybridization, more naming, again, kind of simple alkanes and branch chains, all that. The current plating one, oxidation number, and then we didn't go over just like ester naming. So what I would look up for esters is know how to synthesize them. And so don't forget, right, that's some alcohol plus the carboxylic acid to make your ester. So you may want to look at that, how it's made, how to name those. So that will be helpful. And then the last one is just, it's polymer synthesis. But again, just kind of identifying monomers. And then that is your test. And so the last section, the last 5 are like shorter answer worth 5 points. So your like beefier points are kind of in the middle. So you can, you know, flip through your test, plan. All right, I think that concludes our review. And then if you sent me papers, I have your papers so we can chat about that. Oh, and then check your grades, check your grades, check your grades. So I should have returned everything back to you that you handed to me, although I did find a small pile of papers, although that's actually no one physically sitting in here right now. But just check your grades, check your grades. I think I just got something in from the testing center today, but just, you know, hound me if you need to about your grades. And then I'll keep the percentage column updated. And then if points are funky, I think, yeah, you should have a pile. Like your points, I forgot your bonus points on your essay, but I'll go back and add those. Hopefully, if I don't, send me an email.
All right.
But yeah, hound me. And I will stick around at school as long as people need me. All right, I'm going to shut off the stream, so thank you.